Okay, sir. So, a warm welcome to everyone for our today's session, which will be delivered by Professor Devarshi Gajjar. She'll be talking on integrating genomics and CRISPR for genetic exploration in Pseudomonas aeruginosa. So, before going into the details of our introduction to our today's speaker, just a brief introduction about this science dialogue series. This was initiated almost 1.5 years back, and it was a brainchild from the Gopalji sir. And this platform has brought a lot number of speakers from different corners of the world. They have come on this platform, delivered their lectures pertaining to different topics. They have been delivering their lectures from the different domains of the life sciences based on their own expertise. We have been witnessed we have witnessed the experts from the with their experience ranging from just a new postdoc to the 40 years experienced people who are delivering their talks on this platform. So this is a kind of free service to the participants who are included as a student on this science dialogue series. So they have been benefited from the expertise of these different scientists or researchers or academicians who have joined us, and they have been they have graciously accepted our invitation for the science dialogue series. So, not just wasting any more time, it's time for me to introduce our today's speaker, Professor Devarshi Gajjar, ma'am. So, first of all, it is my personal privilege to introduce ma'am, as I have also worked with in her guidance for approximately 2 to 3 years. I have been under her guidance, so it is my personal privilege to introduce you to everyone. Now, coming to her brief introduction. So Professor Devarshi Gajjar, she has completed her graduation from the South Gujarat University, post-graduation from the Maharaja Sayaji Rao University of the Baroda, completed her PhD from the Sardar Patel University, Anand. Later on, she joined a company which is named as Iladevi Contract Cataract and IOL Research Center, where she has worked as a scientist for approximately 10 years. Later on, she joined Department of Microbiology and Biotechnology Center at the MS University of Baroda in 2014, where she has worked for a lot number of projects which have been funded by state and national funding agencies such as GSPTM, ICMR, SPARC, MHRD. She has contributed very well to the society in the form of her research work, which is being credited by number of articles, which is more than 65, accumulative citation for more than 1,000, and currently she is also a coordinator for the DBT sponsor 2 years post graduation program in biotechnology. And if I talk about her research credentials or credibility, then she has actually presented papers and publications, oral and poster presentation in various national and international conferences, symposium. She has been a reviewer into the multiple journals like European Journal of Clinical Microbiology and Infectious Disease, Three Biotech. These are few to names. And if I say about Gajjar ma'am, that she has played a very instrumental role in various departmental outreach activities, in organizing annual VV Modi Memorial Lectures and national and international seminars and symposium at the Department of Microbiology and Biotechnology Center. She has her research credentials have been recognized by various awarding agencies like American Society of Microbiology, Indian Science Congress Association, Department of Microbiology from the Pondicherry University. So, if further, she has been a regular member for the Institutional Biosafety Committee for the MSU, UGC Care member, committee member, has been a member for the Faculty Development and Maintenance Fund Committee, and many other committees. She has been a regular member at the MS University of Baroda and many other institutions. She has been outreaching through the various scientific activities. So it is my pleasure to welcome ma'am for this science dialogue series, and I hope that your vast academic as well as industrial experience will be definitely benefiting our participants who are eagerly waiting for your talk. So now this virtual platform is all yours, ma'am, and we are keen to listen you.
Thank you.
Welcome,
Thank you.
madam. Welcome.
Thank you.
And I think you are coordinating this builder program also, na? DVT
Yes, I
builder.
am. Yes,
This
yes, I
is
am.
a builder program is very big program in which there is training and other
Yeah,
things happen, and there is
yeah,
a
yeah.
3 crore project now. I think
Yes,
kitna hai?
yes,
three
Okay.
yes,
crore
yes,
project with the that cell and molecular
yes,
biologic associations. Yes,
yes, yes. So
So
it
that
was
is in
headed
the
by Professor Sarita Gupta, but because she retired, then
yes,
I was given the responsibility of
okay.
managing the funds. That
Another feather in your cap, ma'am. That's it.
yes.
yeah.
Already you are doing too much.
It is a
So
long
much.
story actually. पैसे नहीं आ रहे हैं, but anyway, some other day I'll tell that
Ha ha
story.
ha ha ha.
Today, let
Yeah.
me just stick to my talk. So, thank you very
Yes.
much, Poonam, Dr. Poonam, for
Yeah.
such an elaborate introduction, and thank you, Gopalji, for inviting me. And it is certainly a pleasure to talk about some of the work that we are doing in my lab. So, maybe I can start sharing my screen. So, it's full screen, right?
Yes, yes, madam. Yes, yes.
Okay. So today, what I'm going to do is I'm going to share one of the aspect that we are doing in the lab with this organism called Pseudomonas aeruginosa, and uh, I'll be telling you my experience about working with genomics of Pseudomonas aeruginosa and a little bit of CRISPR that we used for editing the genome of P. aeruginosa. So, my lab is an infectious biology lab, and we mainly work on antimicrobial resistance and virulence of these four important pathogens: two bacteria, which is Klebsiella pneumoniae and Pseudomonas aeruginosa, one fungus, Fusarium species, which causes keratitis of the cornea, and we also work on polymicrobial infections, biofilms that are formed on urinary catheters. So we do some metagenomics of urinary catheter biofilms and a few other microfluidic study. But today, what I'm going to talk about is about Pseudomonas aeruginosa because we've been struggling a lot with CRISPR, and I thought it will be a good thing to share my struggles with the audience so that if somebody is wanting or somebody wants to start doing CRISPR in any organism, then can remember some of the tips that I'll be giving you today. Okay, so I'll be covering these three topics first and foremost. I work on antimicrobial resistance, and I always feel that it is my duty to make people aware about what antimicrobial resistance is and what would happen if we don't tackle it properly. So, therefore, first I will tell you about what is AMR and the global health crisis. Then I'll shift to how we do whole genome sequencing and how we try to find the antimicrobial resistance and virulence determinants with Pseudomonas aeruginosa as one example. And then next, taking the leads of these determinants, how we do CRISPR base editing in P. aeruginosa. So let's begin. I'm sure everybody knows what antimicrobial resistance is. It means that the pathogens that infect us, bacterial, fungal, viral, have become resistant to all the available drugs, and therefore these simple infections like urinary tract infections and blood infections, those were treated earlier very easily with simple penicillin kind of drugs or beta lactam group of drugs. Now we are not able to treat them, and therefore initially people patients used to not die of bacterial infections, but nowadays the incidence of death with these bacterial infections is increasing, and therefore it is certainly a concern. Now, of the antifungal, bacterial, and viral resistance, antibiotic resistance is most common for obvious reasons that bacteria have a faster doubling time, and we have a huge repertoire of antibiotics that we use, and therefore antibiotic resistance is more common. Antibacterial infections are also more prevalent, so therefore the kind of drugs we take are mostly antibiotics, and therefore antibiotic resistance is more common when compared to antifungal resistance. And the main reason is that we use a lot of antibiotics very casually than the other classes of drugs. So why AMR is a global health crisis? So this picture I took from one of the pharma industries where they predict that in 2016 you can see the number of deaths by AMR was lesser when compared to the deaths by ischemic heart disease and some of the other diseases. But by 2050 the number of deaths are going to increase tremendously, and therefore this is a global health crisis. Now, the most vulnerable group of people for this kind of crisis are the aged people, old people, and the newly born or the neonates. And the AMR is going to affect these two more than all the others, because the immunity of both of these group of individuals is far less than all the other age groups. So, therefore, both of these group of individuals are going to be affected tremendously. In India, the situation is really alarming. In India, the superbugs are 5 times more than all the other countries, many of the other Western countries, if you can say. And the rate of hospital-acquired infections is 5 times more in India. This group of nosocomial pathogens are called escape group of pathogens, which include Enterococcus, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterobacter. And all of these bacteria cause infections like pneumonia, bloodstream infections, catheter related infections, and all the other infections that are hospital related infections. And if you prescribe the same drug again and again, then over a period of time, half of these drugs will not work to these pathogens because the bacteria evolve resistance. And I will give you some of the examples of how they evolve resistance by overexpressing the efflux pumps and some of the examples. So this again says the seriousness of AMR in India, where you can see the mortality per 10,000 population is in the range of 9 to 10 in India, which is far more than all the other countries, and a little less than the African countries. And therefore, AMR in Asia, India is considered to be very, very lethal, and has to be addressed. This is again the drug resistance index, and you can see India is very, very high in the drug resistance index across all the other countries. And lastly, I would like to touch upon the neonates, and it is said that 1 million Indian children die in the first 4 weeks of life each year. And this is mainly because of the septicemia and the bloodstream infections that the children have to face because of the unhygienic and some of the other conditions of the hospital. Now, what are the drivers of AMR in India? Obviously, overuse, over the counter drugs, inappropriate self medication, poor hygiene and nutrition, poor regulatory watch on the poultry industry, and compromised environment monitoring. All of this has led to the spread of antibiotic resistance in nature. And nowadays, some of the environmental samples also have many, many drug resistant bacteria in them. And therefore, these stressors or the antibiotic stressors are driving the antimicrobes, the spread of AMR in nature. Now, why this is very important is because, as you can see in the last 30 years, this is a slide showing the antibiotic discovery and the resistance timeline. You can see this lower panel shows the discovery of, let's say, the blue circle is for penicillins, the yellow is for tetracyclines, macrolides. And you can see that just after the drug was discovered, the date of resistance is also identified. So each and every drug that was discovered, we have resistance to it. And in the last 30 years, there are hardly any new drug class that is discovered and approved in the clinic. So therefore, this is a matter of concern. Now, why the industries are not investing in this is another important question. But more importantly, maybe the profits are not that good. And another important thing is that even if they come up with an antibiotic, over a period of time, the bacteria are going to become resistant. So maybe that's why the industry does not want to invest in discovery of antibiotics, and so on and so forth. So, to address this, the WHO has given a declaration, and WHO says that we must address this at all facets, and being therapeutics, diagnostics, surveillance, transmission, interventions, and environment. And this, all of these domains have to be addressed for AMR, and we must improve our diagnosis, we must do continuous surveillance, we must monitor the transmission, and we must monitor the AMR in environment, and only then we'll be able to understand and control a little bit of the spread of AMR. The another important thing that WHO does is that it gives out the priority pathogens, which we term as the critical priority, the high priority, and the medium priority. Now, this table is from 2021, and in 2024, they came up with another, like the revised version of the WHO bacterial priority pathogen list. So you can see in 2021, Pseudomonas aeruginosa carbapenem resistance was there in the critical priority list. But in 2024, it has been shifted to the high priority list. That means it has, you cannot say that it has become less important, but you can say that the spread is a little bit not as fast as the other enterobacterials. So currently, Acinetobacter baumannii, E. coli, and Klebsiella are some of the very fast spreading bacteria, and therefore they have been grouped into the critical group, and Pseudomonas was moved from critical to high group. So we started working in 2018-19 when Pseudomonas was in the critical group, and we are still working on it, trying to understand. And in fact, when we collect samples from pathology labs like Toprani and Sterling, and so the medical microbiologist there tells me that why is it that we are not getting Pseudomonas now so often. So that is something very important. I believe that this should be addressed, that 5 years ago we were getting more Pseudomonas, and now we are not getting that much of Pseudomonas. So what could be the reason, and why are we getting more and more of Klebsiella and more of E. coli or Acinetobacter? So these are some of the important questions that need to be addressed. So now I'll start with the next topic, which is AMR and virulence determinants in Klebsiella in Pseudomonas aeruginosa. So what we do is, so something a little bit about Pseudomonas aeruginosa. It is gram negative. It is very fast growing, pigmented, and it can become multi-drug, extremely drug, and pan-drug resistant. There is a range of infections that Pseudomonas causes, and all of these infections could lead to death and could lead to uncontrolled spread of the Pseudomonas in the entire body if you don't prescribe antibiotics at the correct time. So what we do in the lab for both of these bacterial pathogens is that this is the general workflow that we follow, that we collect isolates from the clinicians or from pathology labs. We identify them using 16S rRNA sequencing, and then we also do the MIC and the resistance profile. We try to find the overexpression of efflux pump using the cartwheel EDTA cartwheel assay. And for some of these pathogens, we also try to study some of the immune related reactions like the phagocytosis in macrophages and neutrophils, or biofilm assays. So this is the normal workflow that we follow in the lab. And once we are satisfied with some of these, and once we know that which of the isolates that we have are very important, then we so I am full screen on my laptop, so I will not know if anybody raises a
Yes,
hand.
yes. Don't worry, madam. I will speak or they will unmute and
Yeah,
speak.
yeah.
Yes,
All
madam.
right. Yeah. Thank you very much. Right. So once we have collected certain isolates and we are convinced that which of the isolates are important, then we decide that okay, now let's go further and let's do the whole genome sequencing. So so this is the wet lab we do before sending the samples for whole genome sequencing. Now, one important tip that I would like to give to the audience over here, those who have less money, like I have very less money to run my lab. So, so if you have a lot of money, you can just send all the samples for sequencing and you will get a lot of data. But if you have less or limited money and you want to play around with just sending few samples for sequencing, then your wet lab data is going to be very important. You should have confidence in whether what is the phenotypic characterization of the bacteria, what is the multiplex reaction, what is the relative RT-PCR expression of these resistance genes. And when you are convinced that okay, out of the 50 isolates I have collected, only 8 are important, then you can send those samples for further sequencing. So this is one important tip, that you must be very sure why you want to sequence a particular organism. Only when you get a convincing answer to this question, that why do I want to sequence this organism? What is the unusual phenotypic character that this isolate has that I want to sequence it? Only then send the sample for sequencing. What we generally do is that we sequence it and then we try to find what is there in the sequence. No, I don't think that's the correct way to do it. You first be confident in what the isolate is and why is it so unique that you want to sequence it, and then send for sequencing. Now what we do is we just don't restrict ourselves to short read sequencing, we also do long read sequencing. One of the important reasons why we do long read sequencing is that because in short read sequencing, the genomic DNA is chopped into fragments of 150 base pairs, and therefore, if at all you have multiple plasmids, or if you have genes on plasmids and genes on chromosomes, if you have duplicate genes, then you will not be able to tell whether the gene is on which of the plasmids that the organism have. And this answer will only be given by a long read sequencing. This, of course, is expensive, and therefore we do very minimum long read sequencing, because each long read sequence costs around 40 to 45,000 rupees. Once we have all this data, then what we do is we compile all the genome analysis. And again, you don't need to have a lot of sequences. You can just download all the sequences from PubMed and all these on the databases, and then compile your data with all the other data. For example, I have shown over here, we could compile 245 genomes, wherein our samples were only 8 over here. So, just as I said, the selection of the isolate for sending for whole genome sequencing is extremely important. And for AMR, the categorization of isolate is extremely important. So, you must know the isolate that you are sending, whether it is multi-drug resistant, extremely drug resistant, or pan-drug resistant. And this is the definition from this paper. And I insist that please don't use your own definitions for this categorization. It is all well published, and there are CLSI guidelines. So all we need to do is just follow the guidelines and categorize our isolates into these 3 categories. Once we are convinced, then we send our samples to for whole genome sequencing. Now what we do is we do not send culture. So anybody who wants to do whole genome sequencing here, this is the slide which you would want to copy or would want to take the screenshot, is that we always send our DNA because we want to make sure that what we are sending is extra pure. We do the quality check, and only then we send our DNA. We also tell what kind of paired end chemistry we want. We tell we want a 200x coverage, and per sample we want at least 1 GB data. We always want raw data. We never take the analysis that the company gives us, because we have tried sequencing with so many different companies, including MedGenome, Eurofins, Strand, and there are many companies that we have tried whole genome sequencing, and by far all of them are the same. As long as they give us a good quality raw read data, we are happy. Now, this is the pipeline that we follow for the analysis of all the raw data that we get. We do assembly, annotation, we do the MLST typing, we do the phylogenetic analysis, then we try to find the resistance genes, we try to find the virulence gene, and finally, we also try to find the location of the gene, whether it is on the plasmid or whether it is on the chromosome. After getting all of this data, we prepare an ITOL image, and this ITOL image is important because it will give us a snapshot of all the data that we are putting in it. Now, this is an example of the Pseudomonas data that we have. For example, we only sequenced 17 isolates, but we compared our 17 isolates with 197 isolates genome sequences that were there on the BIGSDB portal. And you can see that there are isolates from Tamil Nadu, highest, Chhattisgarh, Telangana. From Gujarat, there were only 4 when we started the work, Karnataka, and so on and so forth. There was no whole genome data available from the north and the east India. So this was something very important, and we think that there should be equal representation of isolates from the entire country if we want to make a statement about what kind of isolates our country has. Then what we can do is we can also collect the data as per from where are the samples. For example, it's pus or the tissue or bile, ear, and all of this can be done. We also try to find the major sequence type. So MLST or multi-locus sequence type tells us about the evolutionary lineage of the sample. And you can see over here ST or sequence type 357 was the highest in the data that we got from the country, from 197. Now these are oxacylinase, or these are beta lactamase enzymes that we found, and you can see there is a huge range of enzymes that we found. All of these enzymes are present across all these 197 isolates. Some are more, some are less. PDC is Pseudomonas derived cephalosporinase. So again, these are also multiple variants of these enzymes, and they are present across. So if somebody wants to diagnose beta lactam resistance using a real-time PCR or using a normal PCR, it becomes very difficult because each isolate is having a different variant of this enzyme, and that is where we are failing. And till date, the diagnosis of bacterial infection is culture based, and you have to spend 48 hours. First you grow the culture from the sample, and then you do the MIC, and then you tell which drug is suitable. And there are no RT-PCR kits. The only RT-PCR kit that is available is from Biomurix, that is a French company. It is extremely expensive. The kit costs around 13 to 15,000, and therefore it's not affordable in India. So till date, we are doing culture-based methods for diagnosis of bacterial infections. So meanwhile, we also found a new evolutionary isolate, and we submitted the sequence to the data collection center, and they gave a new sequence type, which is 4937. This was submitted by my student, which is written over here, Milan Rathod, and uh, this is just to tell you that this isolate was a little different from the rest. We can do from the genome sequence what kind of serotypes it has, how many carbapenemases they have, and we can then summarize all of this data. Now, one important problem with, so this is how the ITOL image looks like, where you can see the pan drug resistant isolate has this mutation, which is not there in any other isolate, and the susceptible isolate over here doesn't have any of the genes that the MDR or PDR isolates has. So this is a very minor version of ITOL image I showed, because I thought putting 197 dots on one slide will not be a good idea, and you will not be able to see what I want to show you. But this ITOL image actually gives us a snapshot of what genes are there. All these on the top are AMR genes, and all these on the side are our cultures. And depending upon whether they are MDR, XDR, or PDR or susceptible, you can see what kind of genes they have, and which culture or which isolate has the maximum number of resistance genes. So you can see over here the PDR has the maximum number of resistance genes as shown over here. So this is a summary showing the number of genes these isolates have and whether they are there on the plasmid or on the chromosome. Now, one important thing about Pseudomonas is that this ML plasmid or PlaqueNet, this is a tool that we use for Klebsiella, where this tool tells us whether the gene is on the plasmid or on the chromosome. In case of Pseudomonas aeruginosa, there is no tool which can tell us whether the gene is on the plasmid or on the chromosome, and therefore genomics of Pseudomonas is still in infancy, whereas genomics of Klebsiella has reached another level where there are so many tools where you can use MobSuit and you can tell how many numbers of plasmids are there. In Pseudomonas, there is no way you can tell how many numbers of plasmids are there. Now, once you get a lead, or once we got a lead that this is something unusual. So when we found that BLA NDM was on the chromosomal cluster, which was never reported, NDM earlier was only reported on the plasmid, we thought that, okay, this is the isolate that needs to be gone for long read sequencing. And then when we sent this isolate for long read sequencing, I am so sorry, I'm supposed to remove this. I can take this. Yeah. So when we sent this isolate for long read sequencing, we found that this isolate had 2 copies of NDM, 2 copies of OXA, and 3 copies of BLAST-CTXM gene. We also found where it was integrated. Now, all of this maps is not possible with Illumina sequencing. Only when we have long read sequencing, we can tell where exactly the gene is integrated or whether it is present in multiple copies. So all of this data is published. If anybody wants to check both of these papers, we published all of this data. So I can summarize our AMR data. I can tell that whole genome sequencing is an efficient tool to detect all AMR genes and mutations. I have described the limitations of short read sequencing, and I have also told that in Pseudomonas aeruginosa, the tools are not very well built, and there is still a need that we get more and more tools for Pseudomonas aeruginosa characterization and of mobile genetic elements. So now I shift to the next thing of the virulence factors. So Pseudomonas was earlier considered to be more fulminating and more dangerous than the other bacteria, mainly because it has so many virulence factors. So this image you can see, it has enzymes, it has the highest number of type 3 secretion system. It can kill a bacteria, it can kill a eukaryotic host, it can adhere, it can form biofilm, it can form siderophores, and there are too many things that it has, and therefore Pseudomonas was considered to be highly virulent. So I'll just give you an example of one virulence factor, because there is no point telling you the same thing over and over again for all virulence factors as to how we. So once we get the genome sequence, what we do is we align all these genes, and you can see over here. So this is PaO1, which has aspartic acid, whereas all our other cultures have serine instead of aspartic acid, and uh, because of this mutation, all our cultures are, a majority of them are making less exotoxin A. Now, one of the important question is that if it is making less exotoxin A, virulence factor, then why is it so virulent? So this further verification has to be done, that it has a mutation, it is making less of the exotoxin, then how is it maintaining its virulence? Maybe because of the presence of the other virulence factors, but this is what we have got from our genome data. Another study that we did was we compared all the biofilm formation abilities of all our cultures, and we saw majority of them form strong biofilms, very few form weak biofilms. We found that all these cultures have the same growth rates, and therefore the growth rate is not responsible for the strong biofilm. We also did the scanning electron microscopy, and you can see the weak biofilm has only bacteria, does not have the EPS, whereas the strong biofilms has a lot of EPS and lot of extracellular matrix. We did the cell adhesion assay, and you can see in the strong biofilm forming bacteria have more adhesion abilities, whereas the weak bacteria has less adhesion abilities. Likewise, we found that all our weak biofilm forming bacteria are mutants of the pill A gene, and because they are mutants of the pill A gene, they are having this smooth edge, whereas the strong biofilm have the pill A intact pill A gene, and therefore they are having the rough edge, and this was attributed to twitching motility. So if you see this video, you can see the twitching of bacteria. You'll have to see very carefully. You can see bacteria are moving, and by the motility called twitching motility. So the twitching motility was more in strong biofilm, whereas the twitching motility was less in weak biofilms, and all of this is published in these 3 papers that you can see over here. So strong biofilm formation is due to increased twitching motility, loss of twitching motility is due to the mutation in pill A gene. Now, next, what we wanted to do is compare the biofilm and AMR. So we tried to compare biofilm quantification of these 2 isolates. TP10 was susceptible, TP11 was resistant, and we wanted to find what is responsible uh, uh, during biofilm formation. What are those factors that give its resistance? So these were the two important resistance determinants in biofilm tolerance in literature called SAGS and BRLR, which is the biofilm resistance locus regulator. So SAGS is the factor upon activation. It increases the expression of BRLR, and BRLR overexpresses efflux pump. Now this happens, so BRLR will only function when it has cyclic di-GMP and pyocyanin. So when all three of them come together, there is overexpression of efflux pump. So what we did from our genome sequences, we found that TP10 had a truncated SAGS and TP11 had only one variant. And when we compared all the rest of the mutations, they were mostly neutral mutations, so they did not have more of a role. So when we did the pyocyanin quantification, we saw that TP11 had very, very high amount of pyocyanin, and that was responsible for the upregulation of the efflux pump, which we showed that SAGS mutants with high pyocyanin content show increased resistance in the biofilm state. And this, by RT-PCR, we showed that all the efflux pump genes are overexpressed in TP11 when compared to TP10, and this was mainly because of more of pyocyanin synthesis. So increased resistance in biofilm state is due to overexpression of efflux pump, which is in turn due to overexpression or oversynthesis of pyocyanin, and that is what we see. Now I quickly come to the last
क्वेश्चन
part.
मैडम लेंगे, एक क्वेश्चन
Yes,
एक.
yes, 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 Gopalji.
So, Nanar Aditya, he has asked, Good evening, madam. Myself, Nanar Aditya. My doubt is now,
Uh-huh.
how does twitching motility affects biofilm formation?
So twitching motility is because of the pillin protein, which is there on the cell surface. And if the pillin is intact, then the cells will adhere more and spread more. So if you see this image, sorry. So if you see this image in TP8, which is weak, this is a smaller diameter, whereas this one is a larger diameter. So this is a very, so in fact this paper is published in scientific reports, and when we sent these images, the reviewer said that no, this is not enough, you have to send a video uh, uh, along with these images. So then we recorded these images, these videos of the twitching, and we sent to the reviewer, and that's how our paper got accepted. So it's
Adit?
basically more adhesion due to the pillin protein. Yeah.
Yes. I hope you got answer, Aditya. Okay. Please continue, madam.
All right. Sorry, I am actually running a little late. So I come to the last part of my work, which is CRISPR-based editing in P. aeruginosa. Now, because we were finding so many variants in our clinical isolates, we thought that why not do some mutations in the type strain, which is PA01, because there we will have control, because all the clinical isolates have too many SNPs and too many variants. So that is when CRISPR was coming up 3, 4, 5 years ago. Actually, we started 3 years ago, 4 years ago, but it was CRISPR was came up long ago, but we started 3 years ago. So what we decided was that because pyocyanin, so this is the two component system, and these are all the regulators of all the virulence factors. So LASR is an important one, RHLR is another one, and PQS is another one. And both of these control the production of pyocyanin. And because we got more pyocyanin in some of our isolates, we thought let us mutate LASR, or well, let us delete LASR, let us delete RHLR, let us delete PQSR, and then see the effect of these deletions in our strains. Now, deletion was only with the help of homologous recombination, and that is when we read of CRISPR base editing, and we thought that why can't we try base editing, because by base editing we can incorporate multiple mutations in the same strain easily, because people were doing that back then. So we targeted these genes, and so before I move to what we did, I'll just briefly tell what are the currently available CRISPR tools in P. aeruginosa. So either you can do the normal CRISPR-Cas9 gene deletion by double-stranded break. So in this case, we will need two plasmids. One plasmid will have the Cas9, Cas protein, and the other plasmid will have the guide RNA. Both of these plasmids will go in the cell and they will do double stranded breaks, and there will be with the help of neighbor end joining and homology based recombination, we will get the deletion strain. But the problem with this is one is the off target, and that this deletion is not controlled. You might get various levels of deletions. Some isolates you might get more strands deleted and some less. So therefore what people have started doing is that they use CRISPR base editing. In base editing, the advantage over here is that we only have one plasmid. It is called PN-Caspar-Beck, and in the same plasmid we can put our spacer, or we can put the gene for our guide RNA, and the plasmid has the Cas protein. So all we just need to put the spacer in this plasmid, and the Cas protein is already there in this plasmid. So what this base editing does is that it will convert the C to T, and therefore by doing this we will just get one base change wherever we want. So therefore CRISPR base editing is considered to be a little safer and option when compared to the normal CRISPR deletion protocol that we do. So this, I'm sure everybody knows, there is a PAM sequence, there is a guide RNA, the Cas protein gives a double-stranded break, and this double-stranded break could lead to deletion, mutation, insertion. But in this case, we will need two different plasmids. So the red numbers over here are the AdGene plasmids that are available on AdGene. You can just order them. They are not very costly, and the normal CRISPR can be done by using two of these plasmids. One plasmid has the Cas protein, another plasmid has in which you have to put the guide RNA, or the sequence for the guide RNA. Now, what we used is the cytosine base editors. So, a little bit about both the cytosine and the adenosine base editors. The cytosine base editor has the Cas9 and Cas9 protein. So, this Cas9 will not give double stranded DNA breaks. It will just help to adhere or bind to the DNA. There is a cytidine deaminase, and there is a glycosylase inhibitor. The UGI is a glycosylase inhibitor. So this cytidine deaminase will deaminate the cytosine, will convert it into uridine. This UG base pair will be converted into UA base pair, and in the next round of replication, it will be finally converted into AT. Now, similarly, there are adenosine base pairs having the adenosine deaminase. What we used was the cytosine base editors to work. So cytosine base editors convert cytosine to thymidine. What we did was that we converted these codons, CAA, CAG, and CGA, into stop codons. So instead of deleting the gene, we just incorporate a stop codon in the gene in such a way that the protein is not made, and therefore we are deleting the function, or we are stopping the function of that particular protein. So this is a very easy way to stop a function of a particular protein, and people have tried more than 8, 9, 10 guide RNAs in the same plasmid. We are struggling with just one guide RNA. That is another story. But this, according to the literature, this creating stop codons is not very difficult, and you can insert multiple guide RNAs in the same plasmid. So we use the PN-Caspar-Beck for cytosine base editing, and this was created by the NCAS9 with the cytidine deaminase and the guide RNA, which then binds to the DNA, and the C is converted into U, uridine, and the U is then finally converted into, so the C is finally edited into T. Now, why PN-Caspar-Beck? Why did we use it? Because it was available for Pseudomonas. It has the Cas9 protein. It has the ApoBeck, which is the cytidine deaminase, and it has the guide RNA scaffold. Now, because it has the guide RNA scaffold, all we need to do is just put the 20 base pair sequence, which will transcribe into our guide RNA. And this is extremely easy to do. It has the SAC-B sequence. This SAC-B cures the plasmid when you grow the cells into sucrose. So once your editing is done, you can remove the plasmid and again put another plasmid with another guide RNA that you want. So therefore, by doing that sequentially, you can create double mutants, triple mutants, and n number of mutants that you want. So this number that I have written over here is the add gene number from where we had bought the plasmid. These are some of the tools that are used for guide RNA designing. The CRISPR CBI is the older one. Nowadays, people are using ChopChop and CRISPR. I suggest that you use all four and then choose the common sequence that you get. That will be more safe, and it will at least guarantee that the sequence that you have chosen for designing of guide RNA is not very gross or random. This is just a screenshot from CBI designing. It is a free tool. All you need to do is just put your genome sequence, put your target sequence, and it will tell everything, the off target, the guide RNA sequence, and everything will be tell. Once your guide RNA is sequenced and once your plasmid has arrived, that means the culture of the plasmid arrived, now the real work starts. So the first step is plasmid isolation and conformation. This is the gel showing plasmid isolated. This is the gel showing the linearized plasmid. And you can see this is a very high molecular weight. The size is around 10 kb, and therefore it is a low copy number, and it is extremely, the plasmid yield that we get is not very much. You have to do a maxi prep and get a good plasmid yield. Next, when we the spacer is very easy. You just order it from Eurofins or any other Sigma. Spacer is nothing but a 20 or 25 base sequence. You have both the sequences forward and reverse, and what you do is you anneal both these fragments. So you order this sequence, you order another sequence, and you in the test tube anneal both of these, and this step is called spacer phosphorylation and annealing. Once the annealing is done, then we use the gateway golden gate assembly. The golden gate assembly has this enzyme called BSA1, and it also has it is called a one pot reaction, where you put both the plasmid, the phosphorylated spacer, the ligase, and the BSA1 enzyme. The BSA1 enzyme will digest your plasmid and give you a sticky end where your phosphorylated spacer will bind. So this is how it looks. This is my vector sequence, and you can see the sticky ends after digestion with BSA1 has TGGG, and over here it is AAAC. Same is the case with the spacer. It has TGGG and AAAC. So both of this will, this spacer will get ligated over here in the vector. And next what you have to do is just transform this ligation mixture into Pseudomonas or E. coli. Now, once you have done this, you will certainly get 5-6 colonies. I don't think that is a very difficult task to get. Transformation efficiency is very good when you do electroporation. But the real task is conformation of the spacer. So if the spacer is not ligated inside the plasmid, then there is no point going ahead. So the first thing you have to do once you get colonies is either you design internal primers inside your spacer and confirm that yes, the spacer is ligated, or if you have good money, you just send the plasmid for sequencing, and the sequencing will tell you whether the spacer is ligated. Once this confirmation is there, only then you can imagine that yes, there must be some editing happening. So we got a couple of colonies. Now, one of the good thing about our targets was that we were looking for pyocyanin. So immediately when the colony did not give color, it was some indication that
So,
yes,
so there is
yeah,
no way for checking that digestion pattern if
no,
there is
because
restriction. What is the size of a spacer?
25 bases. This is the
If
size of the
if
spacer.
we
So
can, we have some restriction enzyme adjacent, so accordingly we
yeah,
can
yeah. So we
all out
tried
all out
doing
difference
that.
देख सकते हैं ना.
राइट राइट, और वी गेट एन एंजाइम जो सिर्फ यहाँ पे कट करे
हाँ,
एंड प्लाज्मिड को लीनियराइज
या
करे.
दो इंजाइम से दोनों साइड में
राइट,
विदाउट का अलग साइज होगा, विथ का अलग, इस टाइप से वी
हाँ
कैन डिजाइन
हाँ,
सम
यस
रेस्ट्रिक्शन
यस
ऑल आउट
यस
करके.
यस
यस.
यस यस. सो वी ट्राइड डूइंग दैट, बट समहाउ हमारे केस में नहीं हुआ वो. वी नेवर गॉट एनी एंजाइम दैट वाज कटिंग क्लोज टू दिस सीक्वेंस. सो
ओके.
इवेंचुअली हमें पीसीआर से ही उसको वेरीफाई करना पड़ा. ऐसा हुआ. Right. So our target was very easy. Our mutant will lose pyocyanin. So that was initially very easy. Once we get the colonies, we again isolate the plasmid and confirm that yes, the plasmid has gone in E. coli. and then we do a certain phenotypic assays like loss of pyocyanin, and then what we do is we do the curing. So sequentially sucrose ki plate pe grow, when you do, the plasmid will be lost, and eventually the mutant strain will not grow on gentamicin, and that's what your plasmid has lost, is gone now. Now you can put another guide RNA plasmid, and so for example we did with las R, the plasmid with las R, and again we got so many colonies. You can see over here, from all of these colonies we selected one colony which is P1.14, and we did many assays, like we did the protease assay. You can see over here the wild type has so much activity, the mutants have less activity, like P1.14 has less activity over here. Some other colonies had no activity at all. So then we took this one ahead. We did some exotoxin activity. So again, this colony P1.14 had very less activity, and we were very sure that yes, this culture has the edited gene. So we sent it for whole genome sequencing. We also did some Sanger sequencing for both these genes. And eventually what happened is that, so this is the sequencing result. So this yellow region is our spacer, and we had designed to edit this C. It is somewhere around 95 or 96 position C. So we were thinking that this C will become T. So it will become a stop codon over here, TAG. But what we found was that at this position, the G became the T, and this was a transversion conversion, which was never reported in CRISPR base editing. So we are still confused that what went wrong and what happened. So we will again repeat the whole thing, and I still don't know whether this is surely the transversion has happened because of the CRISPR editing, or this was just by fluke that we got. But we whole genome sequenced all these CRISPR base edited cultures that I showed in red. All of these are whole genome sequenced, and none of them showed any base editing that we had planned. So I just thought I should share my personal experience on CRISPR, that we got a bystander editing, and we still have to confirm this by doing again and again. We also compiled the amino acid sequence, and this was a neutral change, like G to T conversion occurs within the codon, and it led to substitution of methionine with isoleucine, and they have different chemical properties and sizing, and substituting methionine with isoleucine could potentially affect the structure and function of LASR and ability to regulate quorum sensing. But we still don't know that whether this transversion is because of the CRISPR that we used, or we have to again do this again and again and be 100% sure. So only one out of the 7 colonies showed the base editing, and this edited one was also a bystander mutation. So I'm still not in a very good position after 3 years of 4 dissertation students and 1 PhD student putting so much of effort and money and time
एक
doing
बार
this work.
करेंगे एक्सरसाइज मैडम, इस पर में आएंगे हम तो डिस्कस करेंगे।
Yeah.
यस।
So no, so I think repeat karna hi, that is the only solution, that if we keep
नहीं,
repeating
आप
and
बोले कि
repeating.
दूसरे जगह भी एडिटिंग हो गया ना?
Right, जो दूसरे जगह हो गया, yes, दूसरी जगह हो गया, वही
Because
हुआ है.
here you have used dead
Yeah,
Cas9,
it
yeah,
is the
not
dead
Cas,
gas. Yeah, so it
which
should not cut. Yeah, it
yes,
should. Yeah, yeah.
लेकिन दूसरे जगह एडिटिंग हो गया because enzyme is bound to that है
Yes,
ना,
yes,
dead cas, so it should
yes,
bind at that specific place as
yes,
per
yes,
guide RNA है ना,
yes,
instruction.
yes,
It should not
yes.
bind. वो कुछ दूसरा रीजन हुआ होगा, क्योंकि दूसरा म्यूटेशन भी तो स्पॉन्टेनियस म्यूटेशन
हो
वगैरह
सकता है, हो सकता है. Right, right, right. So
तो
anyway,
क्या?
I'll just end. To summarize, genomics has revolutionized our study and understanding AMR. So my lab is a very like good for genomics pipelines, and if anybody is interested, I'll be happy to help. That is a short protocol. We train all our MSc students also for our genomics pipeline. CRISPR base editing, yes, I'm still not very confident, but I will not stop doing it because I want to take this challenge, and I want to be 100% sure that yes, CRISPR base editing is also an efficient tool, and we will continue doing it. So these are some of my students. The data that I showed today is mainly of Hiral and Milan. Siddhi, Swati, and Hiral are already PhD and they are doing good jobs. Suraj has submitted. Some of the Klebsiella data was Suraj's. And Pushpendra, Ratika have been really nice to help me. Sonal is my collaborator in the network program, and Dr. Madhvi is also very kind enough to whole genome sequence some of our samples as a part of the network program that we have. I'm also very, very thankful to all my clinical collaborators because it is they who tell us that yes, this culture is very important. If they don't tell us, then sometimes it goes unnoticed. For example, one of the best papers that we published, it was Jyoti Patankar madam from Sterling Hospital. She told that, madam, ये lady मर गई, आप कुछ करो उसके लिए. And that is when we thought that, oh, this is very serious. And sometimes when we are in the lab, we are very much cut off from the real world. So I always feel that being connected to doctors and some of the real, we know some of the real challenges that they are facing. And yeah, this is MSU, I'm sure everybody knows. And I always believe in doing, and learning by doing is the motto in my lab. So yeah, I'll be happy to take questions.